Welcome everyone to the Turing-Roche Tech-Talk series, which aims to share technical resources and knowledge to improve how we do computational-based work, specifically in the medical sciences. If you weren't aware, the Turing-Roche partnership has been established to bring together researchers across academia and industry. And through this, they have several community scholars with different projects. As part of my project, I'm running this seminar series. With that, I will move on to introducing our first speaker. So Mark Adams is our speaker, and he is a senior research fellow in the Centre for Clinical Brain Sciences at the University of Edinburgh, and is a lead analyst in the Psychiatric Genomics Consortium. He conducts large-scale genomic studies of mood disorders, such as depression and anxiety, with a particular focus on incorporating information on variation in diagnostic ascertainment and in disorder heterogeneity into genetic discovery. In this talk, Mark will cover the philosophy and principles behind workflow systems, discuss two systems in detail, which are Nextflow and Snakemake, and share some examples of how workflows have enabled his research.
So a workflow is a system that sort of helps you achieve all of these objectives. So the first way to kind of think about what a workflow is a graph. You've got data, you have code, they go into some kind of computation and spit out a result. And these can sort of get more complicated, that the output of one computation might be the input into another, and then more code is layered on top of that. But one thing that you want a workflow should be able to do is that when the inputs change, it doesn't rerun the entire thing. There should be some kind of caching mechanism that only the bits that change and downstream of that will get updated. So imagine we've got a collection of notebooks. So they're like some Quarto markdown files. That workflows.qmd is the talk I'm giving now. So the first thing is like, okay, you just want to render them in a loop, let's say. So you could write just a little bash script to get a list of all of the Quarto markdown documents in a folder and render them out to an HTML presentation or document. But if you make any changes, like every time you run this, it's going to recompute all of those. So maybe we could sort of add a little bit of logic into that. So we sort of get the base name of the file, and we check, is there an HTML file with that name already in this directory, or is the modification time of the notebook newer than the HTML file? And if either of those conditions are met, then you render the notebook. But this is sort of getting like lots of brackets and stuff. So it'd be nice to encode this into a way that sort of makes it simpler to run and also simpler to expand. So one way you could go about doing this is using a make file. If you've ever installed like source code on Linux or Mac OS, you might have typed this like make install. And this is just a build system that is made for generating a computer program from a series of source code inputs. Like there's a good example in the Turing Way book of using this. So what a make file is a list of output input pairs, and then the command that will produce the output from the input. And sort of an example of that, so I've made a little make file here that will take a workflow Quarto file and produce an HTML output file, and that's just Quarto render, and then the name of the notebook. And if you type make, if you have this make file, and you type make and then the name of the file that you want, it'll sort of match up those rules and then run the rendering process, output the HTML document, and then you're done. But if you run that command again right away, it'll notice, well, the input file hasn't changed, so I won't bother regenerating it, and it'll tell you that the output is up to date. So this is kind of like, okay, we're doing a little bit of reproducibility, and we could also expand this to like, well, we have multiple notebooks, so you just write a generic rule that looks for the .qmd extension and knows that something something.qmd produces something something.html. And then you can write a rule that will generate any of these notebooks. So you can write make symposium.html, make thesis.html, or if someone else sends you a markdown file, you can rerun it and generate the output from that. So another thing that workflows are is pipelines. So this is kind of like a Unix pipe that you run a command and the output of one program can be fed into the input of another. Or you could think of a pipeline as maybe if you've used something like SunGrid Engine or Slurm, sort of the high performance computing job schedulers, you can sort of write relationships between all of your script files that one will wait for the previous step to finish before running, and then you sort of are manually kind of hooking up the inputs and outputs. So here we've got, maybe a little bit of like scalability that if you've got a high performance cluster, you could maybe generate, lots of outputs at the same time. And workflows are also should be parallelizable, so that if you've got a data set, say like you're analyzing genomic data, you could naturally split that up into chromosomes, process each one in parallel, and then combine them all together. So that make file that we wrote before, you can do this using the J command that says run 3 jobs at once, and it'll render all of these notebooks at the same time, given the computational resources of your laptop. But there are kind of some limitations of like doing reproducible science this way. So a makefile has no understanding of the problem domain. It interacts with shell commands, so you can run all of your bioinformatics tools, but it doesn't natively understand the fact that you might want to run like an R script or a Python script or a Julia script. So it doesn't, you can have it call those Python the name of a script you wanted to run, but the tool doesn't have any understanding. It just thinks you're running a program. Makefiles also, there's scalability on your local machine, but you can't automatically say, okay, run this in like a cloud environment or on my institutional cluster. So the sort of the parallelization you can do is rather limited, because each of the steps it's doing also, like some of them might be more computationally intense than others. So some of them might require all 8 cores on your machine or something, and the makefile doesn't know, okay, run this one big part and then run some of the smaller parts separately. Whereas the schedulers, so like SunGrid Engine, Slurm, so that has scalability in that like you can access hundreds of compute nodes at once, but you're sort of manually managing the inputs and outputs between all of those scripts and the dependencies. And if you want to run those things on new data sets, it might be a lot of like manually managing the names of jobs so that they're always waiting for the correct one. And the other problem there is that you set up this nice big HPC pipeline, but it has to run on the cluster it's written for. So you can't kind of develop something on the scale of your laptop and make sure it works and then scale it up to all of your computational resources. So a workflow system is a system that's kind of designed to like package all these problems together and give you a solution for them. So it's giving you some kind of language, sort of like make, you've got input files, output files, and some kind of command that generates those two and hooks things up. So I've kind of broken them into like 3 broad categories. So there are declarative languages, like the Common Workflow Language, CWL. And these are, I haven't actually used them too much, but sort of my understanding is that you describe all the steps, but it's not a programming language. So it's a little bit more limited in the expressivity of it, but the advantage is that what you submit the job to can differ. So there are lots of different like clusters that could input one of these language files and run the analysis. The broadest thing are general purpose imperative frameworks. And what these are is they're running inside the language you're already using. So if you're writing a Python program, you can write a workflow in one of these languages running inside Python. So like SciPype and Dask are Python ones. Targets is an R-based workflow system. But the kind that I've kind of used the most are these domain-specific imperative languages. So they have the expressivity of a programming language, but they're a little bit more general purpose. So you're running them from the command line, but the file that you're writing is actually a program. So it's kind of the best of these two, declarative and general purpose, that you have a language that's tailored to the problem, which is writing a workflow, but it's not tied to a specific programming environment. So the workflow will do all the busy work for you, managing connections and caching of inputs and outputs. It can hook into things like Docker or Conda so that you can describe these are all the software dependencies required to run a particular workflow. And they can also talk to the all of the job schedulers and like cloud environments so that you can scale based on the number of resources that you have access to. So the two domain-specific language workflow programs I use the most are Snakemake and Nextflow. Snakemake is Python-based, whereas Nextflow is Groovy. I assume everyone's probably familiar with Python, but Groovy is a programming language in the Java ecosystem, so it runs on the Java virtual machine. But I think if you're familiar with Python or R, it's pretty easy to pick up. The paradigm of these two workflow packages are actually kind of opposite from each other. So the default in Snakemake is you're writing a rule and you're asking for an output, and then Snakemake will look at your output file and then try to determine what inputs are required to generate it. Whereas Nextflow, you feed it an input, and then it looks at what that input is based on these workflow procedures you've written, and then it'll dispatch that to a particular process to actually do the computation. So each workflow step or rule, it can be a shell command, an R, Perl, Python, Julia script, or you could write a rule or a step that's just native code of the Python or Groovy that you're writing it in. So yeah, and you'll see, I'll show some examples of like the same workflow written in these two packages. So let's start with Snakemake. We're back to the example where we just want to render a Corto notebook. And we write a simple rule. We'll just name it render. And the first part, so render is just the name. You can call it whatever you want. The first thing it takes is an input. And to make it generic, anything in brackets is kind of a wild card. So that can change depending on the file that you want to process. It produces an output, so whatever the name of the notebook.html. And then the last thing is the shell command that will process that input. So the input variable here just gets replaced with whatever the input you've given it, and then it'll run that command. So we've written, so say we want to ask Snakemake to produce the workflows.html, we just say Snakemake workflows.html. It looks at that HTML file, sees that it matches this output rule, and then it'll run the shell command based on the input file that it can find that it matches. So it's sort of looking for these like complementary pairs of inputs and output files based on the wildcard matching patterns that you've specified. It'll sort of run that script, give you some information about what it's doing, and then say it's complete. Whereas if you rerun the workflow, the input file hasn't changed, so it'll just say nothing to be done. All requested files are present and up to date. And this, you can also ask for multiple files. So if you say like, give me all these HTML files at once, it'll dispatch all of those and produce them each in turn. So we're not doing any parallelizing yet. We're just saying, give me all of these different things, and it'll look them all up on your system, find all of those matching notebooks, and then do the render process. So Nextflow is kind of reverse. So the snakemake is, here's the output I want, you find the inputs. So what Nextflow usually starts with are like the parameters that you're giving it. So here, this default one, I'll just call it notebook. By default, it just looks for all of the, it's the shell expansion. So it'll just look for anything with a QMD and process that. Nextflow is written as these workflows. So the inputs become what it calls channels. And this is just like basically a list or array of files, variables, etc. And then it passes those one by one into a process. So here's render. It looks very similar, like that script is the same. The input and output are kind of similar in the sense that like we're doing a little bit more work here to construct what the name of the output file is, but it's essentially the same thing. It's like one input, one output, and the script command to actually produce that. And then when we run it, the thing that we're sort of looking for here is this notebook.workflows.qmd. So that's the input. So we just say, here's our one input file, run this notebooks.nextflow list of steps, and then it'll look at that file, push it through the process, and then make the output. But if you rerun it, it does the same thing. It sees that this file is already there. It tells you it's cached, so it doesn't do any extra work. But yeah, you can sort of see, so in snakemake, we asked for an HTML file, and it ran the rendering step. And in Nextflow, you give it the notebook, and it runs the rendering step. So they're sort of like opposite each other, I guess. And this, I think this kind of changes like which one you would choose for, which reason. We can maybe talk a little bit about that at the end. And then for multiple files, it's a little bit more complicated. You've sort of got to write some kind of shell expansion that matches all of those input files that you want. And it'll see one of them's already been cached, so it'll not reproduce that workflows one, and it will dispatch the other two to the rendering process. All right, so one thing, so what we've done so far is like, well, it's not any more advanced than the makefile example we had. So the thing that these tools like Nextflow and Snakemake are really good for data science is that it can understand script files. So here I've just got like, let's do a simple Lotka Volterra predator-prey script model. So here's a snakemake rule. It produces an output of a PNG. So we just want to run this model and render the output. But instead of a shell command, we're passing it just the name of a script. And it sees, oh, that's an R script, and it knows how to run that. So what snakemake does is it invokes the R script process, but it passes a special snakemake file. So there's the wildcards that sort of match these prey and predator values from the file name, and then like the output file. So what's kind of different here is like there's no input file. We're just actually telling it the output we want, and it knows all it needs is the values from this this file, and it turns those into variables that you can use in the script. So this is just initializing the model with the predator and prey frequencies or abundances. It runs a simulation, makes a plot, and then saves it out. And that plot is the output that we want. So here, kind of encode. So like sort of what's happening here is like the parameters are getting encoded as part of the file name. And it matches those variables and says, okay, the script has said this is a variable, so it kind of knows what to do with it. And then produce that file if we were to open it up. In Nextflow, instead of asking for the file, we're giving it the inputs. So instead of feeding it in a file, we're just giving it some values. And this is one thing that kind of makes Nextflow different, is that it can pass values and not just files between the sort of steps in the workflow, whereas Snakemake is very file focused. But it's sort of a similar thing where we're sort of constructing these channels, which are just like the list of parameters that we want to use. They get fed into a function that represents what the workflow step is going to be. So those values, predator and prey, are the inputs. But the difference here is in Snakemake, the script was an external file and was sent to a specific object. In Nextflow, the way you usually do it is embed the script as actually part of the Nextflow script file. So the shebang R script is just telling Nextflow what program to run the rest of the script with. And then it uses the same variable substitution that you're familiar with in Bash, or you've seen in the previous rules of just inserting those values into the script and using them to construct the file name. So if we were to run that, it would sort of produce exactly the same kind of file. Okay, so the next thing that these workflows do is working with resources. So this is where you're telling the workflow each step in the process, like how much of a slice of your computer or how much memory do you want to do. So I'm just doing some commands from the Plink, which is a program for working with genomic data. So in these two rules, I've specified like the number of threads and the amount of memory that's required. And then those are getting inserted into the commands. So Plink has like a threads and a memory command. So you can just tell it multi-threading, use this many threads, and don't use more than this amount of memory. But where this becomes kind of powerful is once you are ready to run the script. So you specified how many CPU cores and how much memory each process requires. And then when you run the snakemake command, you're going to tell it, so the C4 argument is use 4 cores. So if I had a 12-core machine, for instance, the first step is the simulation, which is pretty quick. And the next thing it runs is a principal components analysis, which is a little bit longer. And if you say 4 cores, it'll run like those first 4 jobs from the simulation, then run the next 4, and then it'll say, okay, that PCA step requires 4 cores, so it'll dispatch each of those in turn. So here's just a plot kind of showing like the occupancy of those 4 cores over the course of running this workflow. But if you had 12 cores, it would dispatch all of those 8 simulation steps all at once, and then it sees, okay, the next thing I need to do are the principal component analysis steps. Each of those takes 4 cores, so it's able to run 3 of them at a time. So that's kind of like scaling up to what your laptop can do, but always fitting within the bounds of the resources that you've told it to consume. So it's doing all this kind of managing of like looking at the available resources, looking at how many resources each of the steps takes, and then distributing things to run to parallelize as much as possible. All right, so the next thing that these workflow programs can do is interact with job schedulers. So this is sort of like scaling beyond your laptop. So if you're running cloud platforms like Amazon Web Services or Google Cloud, or if you have a local institutional cluster that might be running SGE or Slurm or any of these other ones, I'm not even actually sure what they all are. Snakemake has profiles and plugins that can interact with these, and Nextflow calls them executors. And there's also a list, there's a GitHub repository for Nextflow that actually has the specific configurations for a lot of institutional clusters. So you might want to go see if your local cluster is there. And what that does is encodes knowledge for like how many jobs is a user able to submit to the scheduler at once? What's the maximum amount of memory that's like among all of the nodes available? Like what's the max memory, and it just sort of encodes all of that information. And then accessing that is sort of as simple as adding this like profile. You install the SunGrid engine profile for Snakemake, and then you just tell it, hey, I'm running on SunGrid engine, and it'll know how to rewrite and link together all of the steps of the workflow to dispatch to that scheduler. But this also makes it portable because you might have a collaborator working with a different system, and they can just install the profile and information for their system and hopefully run your workflow like unmodified. So finally, the last part of portability is software environments. So both Snakemake and Nextflow, sorry, have built-in support for Conda, Docker, and Singularity containers, as well as the module command that's available on a lot of HPCs. And what you do is you just sort of tell, like as part of the rule, what the different software requirements to run this rule are. So for PCA, I can invoke a Conda environment. I could list a container that has Plink installed, or I could list the module that's available locally on my cluster. And when you run Snakemake, you just say, okay, run this with SunGrid engine and use Conda or use a container, and it'll look up exactly what it needs. And it's also possible to not like hard code these. This could come from a configuration file, for instance, to sort of like make the rule set like as generic as possible. And then it's just substituting in like what's available. Okay, so that's kind of it for like a broad overview. And then the next thing I'll just kind of go through some examples of how I've used that, but it might be a good place to stop for like any kind of questions about those, sort of a little bit of the hows and mostly the whys of using these programs.
I do have a question actually about this slide in particular. I think I'm just not entirely sure if it's like this Conda YAML, is that an existing Conda environment, or is it telling it to build?
Yeah, it tells it to build it. Yeah. So here, if you pass it a YAML file, Snakemake it'll sort of make a private Conda environment with the software you specified. If you already have a Conda environment set up on your computer, you can also just pass the name that you wanted to use.
How does that work if you are using, like, maybe you come into more data, so you're running it on a new set of data? Will it kind of cache the environment that it's built, or will it be building it every time?
No, yeah, it'll save that environment. So it only builds it the first time that you run the workflow, and then it'll just keep that saved in like a private directory inside the like next to where the rules are all defined.
Okay, cool. And then I'm not sure if you saw the chat, but someone has asked, Otto has asked, does Snakemake support running rules where there is no output file? For example, the process uploads data to a database.
Yeah, that's possible. Yeah, so there can be no actual output file produced. You usually sort of have to like, I think what I end up doing with Snakemake is you make kind of like a dummy. You just like touch a file to be like this command was run. So it knows, but there are also ways of like you could write a rule that like checks the data, like was this data uploaded to the database about whether it executes or not. So yeah, you can have you can have rules that are just like evaluate an expression to see whether something's been done or not.
Okay, cool. And then also Thomas Kylie has asked, when running using Slurm, for example, do you run the command on the cluster, or can you run that locally and have it submit the job?
Usually you're running it on the cluster. There are a few of these environments that do have dispatch to, like, you type the command locally and that dispatches to the externally, but I think that's usually limited to like Amazon Web Services and Google Cloud, where there's like a kind of API that you're interacting with. I've not seen anything for Slurm where you can like type the command locally on your laptop and it'll dispatch it up to the cluster. But it's possible that there's like a plugin that'll do that, but I don't think it's built in.
I'm also not sure if maybe like I just misunderstood the question, but like my understanding of Snakemake is you're running it on the cluster, but it also submits Slurm jobs.
Yeah,
So
I think wherever the question is like, do you need to run the Snakemake program? Yeah, so Snakemake it'll continue to run. So like normally what I do is like you log into an interactive node and then you start the Snakemake process and it'll keep running, dispatching jobs to the background. And then once all of those jobs are complete, it will exit. So yeah, the Snakemake or Nextflow process kind of needs to run like interactively in the foreground so that it can keep track of what the backend cluster is doing.
yeah, and you can use things like tmux or screen to make sure that even if your SSH connection breaks, that process is still running. Someone's also asked a question that, well, they've said, Kai Lim has said, thanks for the great presentation, Mark. I mean, I think you've got a bit more to go, but may I know what you find the most challenging and frustrating in using Snakemake and Nextflow?
So Snakemake, the thing I find most frustrating about Snakemake is when a rule like breaks and like stops in the middle. It's a little bit harder to get like the workflow into a state where you can kind of debug it. Whereas Nextflow has a really nice thing where it kind of packages the rule and the script all up into like and all of the input files into this little self-contained subdirectory that you can just like change to that directory, run the program like as from the shell, and it'll just run the thing that's trying to do there, and like you can sort of interact with it. Whereas Snakemake, you've got to kind of like read back in the script and be like, okay, it sometimes kind of outputs the shell command, but then it's like, oh, I've got to reactivate the conda environment and figure out exactly, and it's all kind of running it more in the directory where it's working. But frustrating things about Nextflow is the fact that it's groovy. So it's a little bit easier to work in Snakemake because like I know Python pretty well. So I think the grooviness, like I've got to do a lot more Googling for Nextflow just to be like, how do you do regular expression parsing in Nextflow? And then, yeah, I think, yeah, I'd say those are kind of the two main ones. All right, so maybe I'll go through a few examples of like actual workflows I've written and sort of like, kind of what the big moving parts is. So the first one is one of the main things I do are genome-wide association studies. And I work with big data sets like UK Biobank. They're usually multi-ancestry. So that gives kind of a natural partition of the data set. You want to repeat this analysis for each ancestry cluster. So here I've just kind of made a simplified diagram of the workflow that we've got this list of ancestry clusters, genotype data. It does QC for each cluster, does a genome regression step, and then passes all of that into a genome association step that is parallelized over all of the chromosomes. And then you could like have a meta-analysis that these all feed into at the end. So I've written this workflow in Nextflow, and like the first thing in the file are basically all of the possible input parameters. So it'll do different things based on whether it's a binary or quantitative phenotype. I've got a bunch of lists of individuals to like include or remove from the analysis, a list of all of the genotype files, covariates, and then like sort of some meta things of, say, if you're doing a case control association study, you don't want to analyze a phenotype if like there are too few cases. So it sort of does some filtering of the inputs to make sure that it like meets those requirements. Both Snakemake and Nextflow have ways of processing like a CSV file. So you can feed it things that it's like, I want you to process each row of this file separately. So here I'm doing things like I've got a list of every participant in the study, which ancestry cluster, and here's a little bit of Groovy code that will read that file in and then find like, what are all the unique clusters? So it's like European, African, East Asian, and just identify what those are. And those become the basis of the parallelization over those clusters. But these could be anything, like instead of ancestry clusters, you could do like male, female, and it would treat it the same. So the first thing is it just sort of combines all those first sets of input files and then runs them over each cluster. So there's this each here that'll just be like, whatever this script is, whatever it does, just do it for each of these, and it'll parallelize that automatically. The other thing is like whether you are doing a genome-wide association study of a binary or quantitative trait, like the software might need different flags. So it's like if you're analyzing a binary trait, you need to pass certain flags to the software of like, oh, calculate allele frequencies for cases and controls, or maybe use these different thresholds. So each of the kinds of input files have to be treated differently. So what this script does is it just sort of makes these lists of like the MDD phenotype file is binary and the body anthropomorphic measures are quantitative. So it's sort of like this script will sort of tag those with these keys. And then it uses those keys to line up like which flags, if it's a binary file, use these flags. If it's a quantitative file, use these flags. And then from then on, the rest of the pipeline can be run the same because like each input phenotype file sort of has this meta information and needs to be passed on to the actual genetic program to do the analysis. So it's things like thresholds and like computational options that only apply to one kind of input file. Yeah, so the next step is just like merge all these things together and then push them into the first step. But here's where we get into kind of the resource usage. So I've got some like use 8 CPU cores. The memory is dynamic, so it'll look at like how many phenotypes are we trying to analyze at once and allocate more memory if it needs it. Or if the task runs out of time, it'll like keep on trying with more and more time. Maybe the sample size is like really big for some of these. So there's sort of these dynamic things you can do that you don't have to hard code exactly how many resources a particular step can need. You can base it off information that's associated with each of the inputs. And then parallelizing across chromosomes is also really similar. You just be like, okay, here's a list of 23 chromosomes. Here's how they get fed into, like the command is like a subset by chromosome, and it'll just do all of that in parallel automatically. And then you write a rule to like how all of those things get merged back together. So it's sort of like simple to follow the logic, even though each step might be really complicated. You sort of have this nice list of like, do some quality control, do the first step of the analysis, do the second step of the analysis in parallel, merge things together while keeping track of, all of the like who to keep in each part of the analysis. So only do this ancestry group. It kind of keeps all of those input files like in this nice little bundle as it passes through, so that you have the output files that make sense at the end. And here's like just a good example of like the multiple levels of parallelization. So this workflow is written in such a way that you could pass in like 10 input phenotype files, and each of those files might have like 10 phenotypes in them. So you can sort of be analyzing like hundreds of phenotypes all at once without needing to modify any of the code that you've written, because the workflow is able to like understand how you want all of those things to be parallelized. So yeah, it makes it really easy to like scale this up to larger data sets, and more inputs. Another workflow that I published on my GitHub is processing UK Biobank data releases. So this is like just so many just different steps you got to do. Like you got to decrypt the file. You might want to like add in some metadata about, like you might want to produce a data dictionary for each of the new sets of data releases you've had, like for reference. And you also might want it to be like in like a nicer to work thing. So like I've made something that'll take a UK Biobank release and turn it into a DuckDB database file that makes it easy to access from a R or Python, even though you might have like more data that can fit into RAM, and you can like access it easily. So it's just sort of, I got tired of like doing each of those things step by step. So now I've just written a workflow that I only need to provide those two input files, and I know it'll produce exactly the output that I want. Oh yeah, that's a Nextflow. And then Snakemake, most recently I've used it for a meta-analysis of major depressive disorder. So this is something where I've got to meta-analyze like dozens and dozens of genome-wide summary statistics. So we'll do all this like formatting, QCing, meta-analysis, feeding that meta-analysis into downstream things for like fine mapping, gene finding, and then producing a notebook where you've got like text you can copy and paste into a manuscript. And like the notebook like understands what all these input files is, so it can give you, sample sizes and descriptive statistics and all that kind of stuff that is a little bit of a pain, like when one file changes, knowing what in your manuscript also needs to change. So it's possible to kind of link all of those things together, even though it's this huge like cluster-based, gigabytes of data. So just an example of like the snakemake rule. So I feed it this configuration file where I've got these genome-wide summary statistics, and they're all named like all kinds of different crazy, weird names that don't make any sense. So they all get reformatted into kind of a common structure where it's like the format, the cohort, the genome build, and then some kind of reference for like which version of this cohort's sum stats does this belong to? So you can kind of like keep for like, so that's kind of a harmonization of file names. That's what this step is doing essentially. And then that makes it easier to like have a common referent for how all of these files fit together. And like here's an input rule at the end. And it's, so rather than like having to tell it what files I want, it just reads it from the configuration file and will produce, given the configuration file, it'll produce all of those automatically. All right, so yeah, so I guess just kind of a conclusion there. So, sort of what the reproducibility, replicability, scalability, and portability are in terms of what a workflow. And the code for this talk is on the link there. And then there's also this really nice GitHub page with just a list of like what all of these different workflow packages are. There's just dozens and dozens of them. So hopefully if you're interested in this, you can find one that suits the sort of needs that you have. Okay, and I'll end there and just take any final questions and open it up for discussion.
Great. Thank you, Mark. That was a great talk. Super interesting. And I hope people got a bit of a sense of like how using these can actually make your life a lot easier. You don't have to remember all these commands all the time. They're recorded somewhere and they are like linked together. So as you said, you can just like change one thing and it'll all know to like run those things. So yeah, we do have time for a few more questions. If anyone has any, they want to pop them in the chat or ask. Now is your time.
I'll also expand a little bit about, I think one of the questions about things I find frustrating, so I could say a little bit about what I like about each one. So SnakeMake is much easier to get started with, because I think you've got like rule, input, output, script, and that kind of makes it easier to write. Like, yeah, I feel it takes less typing to kind of get started with SnakeMake. So I think I kind of like it for that reason. Maybe the problem with SnakeMake is like once the rules get really, really long, it's hard to have a model of like how they all fit together. So I like Nextflow. You've got this separate workflow block that kind of describes from start to end the input goes through a function and then like comes out. So I think it's a little bit easier to like read it from start to end. Okay, see a question from Thomas. I don't know if you want to read it out or.
Yeah, happy to. So Thomas was saying, have you used either of these with something like DataLad for associating changes to data with specific commands?
Not familiar with that, so I don't really know what it does.
Fair. Yeah, it's someone else linked it to me at the same time I was learning about SnakeMake, and they seem like a really useful combination of things. So the DataLad seems to be like you like run a command through DataLad that's like some script that processes some data into some new data, and then it kind of uses Git in the back end to like say, well, this data file came from running this command. So you kind of have like almost like a logbook of how you got the data that you got. And it seemed like it would work well with this. So I just wondered if you had tried it, but no worries if not.
Yeah, I've not tried it, but like SnakeMake and Nextflow can kind of generate a report. So as it's run, it'll kind of it can kind of give you a logbook of essentially the connection of like this input file was run through this command and produced this output file. Yeah, but I guess it's also kind of like yeah, so I guess one difficulty in SnakeMake is if you make a change to the script, having it like kind of know which version of the script produced like a particular output file. So I think as you're developing things, that can kind of become a difficulty. Whereas Nextflow is really good about caching the actual script that produced a particular output. So that if you like so Nextflow is really good at like if you're using version control and you've run the analysis once with one version of the script and you make changes and run it again, it'll keep those like the output files separate from each other. And if you roll back to the previous version of the script, Nextflow will be like, oh, the script is unchanged. I've seen this version of the script before, and I've already cached the output file. So it's really good about like keeping its own sort of private knowledge base of like, yeah, those things. Whereas SnakeMake's a little bit more file based, so it's like you somehow have to kind of encode in the file name which script was used to produce a particular output, and that can maybe like be a little bit more difficult.
Yeah, I have a follow-up question from what you just said. So I know with SnakeMake, like if you change the actual snake file, it will realize like if code has changed and it might trigger a rerun. But if you are calling a script and that script changes, does SnakeMake realize it's changed, or do you have to kind of like force it to rerun?
Yeah, so what I do usually is if I've got a script like that, I make that one of the inputs. So then SnakeMake will know to look at the script, and if it's changed, it knows that that rule needs to be rerun.
Okay, cool. That's a good tip. Does anyone else have any questions? Okay, well, that was very well timed, I think. So yeah, just thanks everyone for coming, and obviously thanks, Mark, for that talk. That was really useful.